the challenges as well, and what is realistic and what is feasible, for patients, especially. But it will be a general discussion, and I will be directing the questions to all of you and each one of you separately from your own perspectives of your field. So you will have some leeway to, expand on certain topics that you think are more important.
Perfect. Thank you.
Thank you.
And of course, we will also have some questions from the audience that I will be keeping an eye on during our discussion. If something comes up that is quite interesting, then we can fold it in. Hello, everyone, and welcome to this Falling Walls Circle table. This year, our life has been very much in a virtual setting for many of us. And although this has forced us to be physically apart, it has also given us many opportunities to interact in exciting ways. Today is such an opportunity, at least for me. I'm happy to welcome all of you from New York City, and I'm joined by our esteemed panelists from Germany and Belgium. Before we start, I would like to remind our audience that you can use the chat function to send us your questions, and we will do our best to get to them during the course of our discussion and at the Q&A session in the end. I would also like to remind you to keep an eye out for our daily highlights. Every day at 1 p.m. Central European Time, Falling Walls and Berlin Science Week present the highlights of the day. Both as preview and in retrospect. And please also check out the next Falling Walls Circle table, which is tomorrow at 12 p.m. And the topic is the future of computing. Is it a magic wand for innovation or a threat to humanity? But now let's move on to the topic of our discussion today, which is whether cell-based interceptive medicine can revolutionize healthcare. So I'm very happy to our panelists today, Professor Stephanie Demmeler, who is the director of the Institute of Cardiovascular Regeneration at Frankfurt University. Hello, Stephanie.
Hello.
Professor Angelika Eggert, the director of the Department of Pediatrics at the Charité in Berlin. Angelika is a physician specializing in pediatric oncology and hematology. Hi, Angelika.
Hello.
Professor De Strooper, who is the scientific director of the UK Dementia Research Institute and a professor at the KU Leuven and VIB, and is an expert on neurobiology and neurodegenerative diseases. Hi, Bart.
Hello, nice to see you.
And Professor Nikolaus Rajewski, who is the scientific director of the Max Delbrück Center for Molecular Medicine and an expert on single-cell technologies.
I have to, I'm sorry, correct this though. I'm not the director of the Max Delbrück Center, but I'm the director of the Berlin Institute for Medical Systems Biology at the Max Delbrück Center.
Oh, I'm sorry. Yes, that's a small but very important detail. Apologies for that. Okay, so moving on to the actual topic of our discussion, we know today that a central pillar of modern biomedical research and medicine itself is the effort to understand the function of human cells during the healthy state, and also what changes in a pathological state leading to disease. And this has led to huge progress, especially in the last decade, in terms of our understanding of the biology of tissues and cells, and has advanced our ability to diagnose disease, to treat disease, especially when it comes to molecular medicine, biologics, small molecule treatments. However, treating diseases is very complex and difficult, especially diseases that are broad, such as cancer, cardiovascular disease, neurodegenerative disease. So we know that we need to devise new ways to understand the underpinnings of disease, new ways to monitor the progress of disease, to diagnose it, and of course to treat it. So an exciting way to do this is through cell-based medicine. And this encompasses the spectrum of biomedical research itself at the cell-based level, to cell-based therapies aimed at specific diseases. So today we will discuss about the research and technological advances that are providing the potential to achieve this, and whether this potential can be translated realistically into revamping the biomedical process and benefiting patients in the shorter term or in the longer term. So let's start our discussion first by maybe what do we mean by cell-based interceptive therapy? What does it mean for each one of you in your areas? Perhaps Stephanie can give us her perspective.
Thank you very much. Of course, the cell is the ultimate unit of life, and as such, it's very important. So this field covers many different aspects. Maybe I can start with maybe one of the obvious you may think of, and this is cell-based therapy, because this is, of course, one of the things which would be therapeutically applicable. And this has been used in the past in many aspects, but we are far from yet in the cardiovascular field using this. But in other fields, with I think we will learn more from Angelika and the others, there have been progress made. And I think this is the first aspect I want to mention. I think the other people will also add some of the aspects which we feel important in the field of cell-based interceptive medicine.
Thank you. Angelika, maybe you can give us your perspective?
Sure. I think in the cancer field, the cell is in the center of everything. So the disease starts from a cell, and we need to know which are the important cells that give rise to cancer. So in the past, we have only been able to focus on tissues and characterize whole tumors. But with the recent developments, we now can focus on the cell or even the subcellular level and describe what is wrong, maybe at a very early time point in the future, not only in the tumor tissue, but in the blood of the patients. And from these technological developments in diagnostics, we hope to be much more precise in our definition and identification of the origin of each cancer. And if we can very precisely identify the cells causing a cancer, we can, of course, also be more precise in the treatment options. And the treatment options are, of course, in the cancer field manifold. Chemotherapy is very broad, but there are now molecular targeted therapies. There are cell-based therapies, for example, CAR T-cell therapies, TCR-based adoptive T-cell therapy, and all these therapies can directly be targeted to the single cells under the precondition that we identify which are the cells that we need to target. And this is a totally innovative field opening up in the recent years, and it's fascinating to see that and to participate in this.
Thank you. Bart, what is the status of cell-based medicine in neurodegenerative diseases?
So really, I think this is a field where I expect really a revolution with this new technology. It's what the problem was with brain is that every cell is basically different, and it's also very difficult to get brain chunks like in cancer to do any analysis. But with this new technology, we can really look to a few thousand cells, 10,000 cells, which is really a small number, and we can almost individually say what the cell is doing and how it's changing. And for instance, for Alzheimer's disease, it's changing the way we think about the disease. Before we had this focus on dysfunction of the neurons, the neuronal circuitry, but now thanks to this technology, we can look how the other cells, the support cells in the brain, react during the disease. And it's very clear that Alzheimer's disease, to give one example, is a very complicated cellular interaction disorder where gradually you get a decompensation of the system, and it takes about 10-20 years to evolve from the initial biochemical changes, which we could measure before, towards the full development of dementia. And this is the cellular phase of the disease, and this consists of these complicated interactions between the different cells. And so we are now really able to measure that and to detect that step by step. And so there's also other fantastic ideas on the horizon, especially, for instance, in the Parkinson field, where we know that specific neurons are getting lost. And with these new approaches and new technologies, we are able to induce either astrocytes, one of the support cells, to become a neuron, or we could transplant stem cells differentiated into neurons to support the brain and to replace part of the losses. So I think it's really a complete revolution for neuroscience, which we urgently need because we really got stuck with the classical approaches to treat those disorders.
Mm-hmm. Thank you. And I think this is a very good lead-in to Nikolaus and your expertise. So as an expert in single-cell technologies, what is your perspective on cell-based interceptive medicine?
Well, so I can speak maybe also for the Lifetime Consortium, which I'm one of the coordinators of, which consists of a few hundred researchers in Europe, in 17 European states or 18, who have come together unified behind this vision that we want to implement in Europe cell-based interceptive medicine in the next time. And we have actually worked out a detailed plan how to do this. The key point of the technology you mentioned, the single-cell technologies, is that it allows you to look at to detect deviations in molecular deviations in cells from the path of healthy, let's say, much earlier than you could do with previous methods. And this means that Bartel Stroper just explained it, and Angelika also said it for cancer. You are able to have a grip on the disease much earlier than normally when you go to the hospital, where everything is much more evolved and much more difficult to treat. So the hope is that with these technologies, we can actually intercept. That is, we can pull the cells back on the path of health, if I may say it in this loose way. The reason for this is that if you detect aberrations early, then understanding the molecules inside the cells, which is what we can do with our new, we can count the molecules inside single cells now for many cells in parallel. In other words, we can find out about the mechanisms that go wrong early on in a disease. And then, of course, we get ideas and precise ways how to intercept, how to bring these cells back to health.
Mm-hmm. I think this is a very interesting point. And I'd like us to discuss a little bit how you view this going from the biomedical research side to the medical side, to the patients, because one aspect is, of course, understanding the disease, the underpinnings of the disease, and studying this so that we can devise therapies, inform treatment options. And of course, the other side is I'm a patient, I go to the hospital and doctor, and we can use approaches to detect the disease earlier and give me treatment options. So how do you view this? Let's start first from the cell-based biomedical research side. You know, what Angelika was also touching on before, understanding what is the cell of origin, for example, what cells are involved, what are the technologies and the methodologies that are cell-based that are helping in this manner. Perhaps now that I mentioned your name, Angelika, you could give us your perspective for cancer, and then maybe we can move to someone else.
Sure. I think there are two different aspects that help us in medicine. One is on the diagnostic side and on the disease monitoring side, and one is on the treatment development side. If I start with the diagnostic side, I think the new information that we can get today are very important to identify the function of cells in their spatial context. And what we can do today is characterize tumor tissues on the single cell level, but what is under development, and groups internationally and also in Berlin have already started to use the technology, is a spatial view on the cells, on plasticity of tumor cells, so their phenotype, their transcriptomic profiles in the 3D, in the spatial context of the tumor, giving the cells a specific function. For example, if a cell is localized close to a vessel, it has a different meaning for early dissemination and metastasis potentially. Or if a cell is located close to the invasive rim, that means something different than being in the center of a tumor. And we can collect this information and identify new ways of specifying the cells that we have to target, and that are drug targets for development of small molecule drugs, also of cell-based therapies. So this is we identify a new target for therapies, but also if we know what to monitor, we can implement new diagnostics, for example, based on liquid biopsies. So up to now, we are used to diagnose a cancer by taking tissue biopsies, but in the field of liquid biopsies, taking blood samples, a lot of technological developments have been made in the past years, and we are now able to follow a disease and to monitor minimal residual disease, so the rests of the cancer in the course of treatment in the blood samples of a patient, and this is much more sensitive and maybe much earlier to diagnose responses to treatment, but also relapses of cancer. So there are many fields in diagnostics, but also in therapy that can be informed by these totally new information that is gained from the technologies, but also from artificial intelligence, data science approaches that are needed, of course, to analyze these huge volumes of data that we can now obtain.
Bart, you mentioned previously how difficult it is to study processes in the brain, and also how in recent years it has become apparent that diseases like Alzheimer's are diseases of the environment of the brain, right? And cell-cell interactions, not just the neurons themselves. How are cell-based methodologies helping us understand the disease better and also hopefully develop models and treatments for the disease?
Yeah, it's really a revolution which has started in our field. I would like to stress also in the discussions about this that the translation in the clinic will need time, and we need to get the chance as scientists and medical doctors to apply those techniques and to explore how far we can go with it. But what I see now already is that a disease where we were looking at a very molecular way, a very simplistic model where you have amyloid in the brain accumulating, and then neurons react on that and they start to be disturbed and the signals change. Now we can just take small samples, even from during operations, where we take a small amount of cells, and we can really depict how these different cells interact with each other. And we can now show how inflammatory cells are involved and the blood vessels, and how the neurons and the astroglia over time change their response on this amyloid. And this is a process over 20 years. We can now do that because we need only very small samples to do that type of analysis. And so we can really see in the different phases of the disease how the brain is coping with the problem. And I think it's one step further then to understand, when we understand this process, to identify the molecules which could become the new drug targets for the disease. And this is really something extremely important for the field, where we need to diversify our portfolio. And so that's happening at the moment. I can go more in technology details, but that's maybe not the goal of the meeting here. And so I think the promise is, in the first place, in bringing us a complete new gold mine of targets. That's the first promise of this approach. But the second, and that's at least equally important, is that we will be able to stratify our disease now. So Alzheimer's disease is a big category, and most people think that it means dementia. I think it's absolutely bad to put these neurodegenerative diseases in that basket, because that would be like we would study cancer still in the end phase of the disease, when the people are cachectic and metastatic, and everybody knows that this is really a very late step in the disease where you cannot do much anymore. But for Alzheimer's, we are still thinking about curing dementia. And so with this cellular thinking, this new way of thinking about it, we realize that there is a lot of things which are much more subtle, which are not seen in the decompensation of the brain, which you see in the clinic, but which are much more subtle, happening decades, literally decades before you enter. And so with this single cell type of analysis, we have these precise tools allowing us to follow that. So it's very clear that this will translate in diagnostics. And to give you one example, you could take cerebrospinal fluid. Now we do all kinds of biochemistry on it, but you could really take the cells which are in this fluid and do analysis and see how certain responses are changing before you have any clinical phenotype. And I hope really, but that's probably a little bit too much in the future. We need to do that. We could take also blood cells and see whether there are neuronal cells or microglia or any brain cell circulating, and we could analyze that and decide whether we are on the way to a neurodegenerative disorder or not. So I think both therapeutically but also diagnostically, the promises are extremely big, and it will take time and investment to develop those. But if you don't do it, then it will be a really missed chance. I'm thinking about gene therapy, which started 20 years ago, and I still think if we would have put much more effort in it and much more belief in it, while we are now only capturing the first fruits, we could have done that 10 years earlier. And I think it would be really such a pity if we don't do that for the single cell approaches, which have a huge potential.
Yeah, absolutely. Switching a little bit to a completely different disease, cardiovascular disease. Stephanie, what is your perspective on cell-based medicine in your field?
So maybe I can add some aspects to what we are just discussing using single cell technologies. And maybe I can give you 3 examples in cardiovascular medicine. Of course, we have the problem as people in neurodegenerative disease have, that the heart is not easy accessible. So you just can't get a lot of tissues. It's not like with tumors, which you cut out and then you can look into the tumor. But we can get small biopsies without harming the patient so much. And of these small pieces of tissue, we now can learn tremendously, because we can separate each single cell and can look into the composition of the tissue, how the cells invade the heart, whether they are aggressive immune cells. And I will give you 3 examples how we now use this technology. The first is a study where we're looking into kid hearts. So children with pediatric cardiology, cardiomyopathies, and in these hearts, we can get samples out of these children. And then we can look what is wrong with the hearts of these children. So we can look on a molecular level how the signaling pathways are disranged, what cells are reacting. And we found interesting differences already between very young kids and kids which are a bit older, and maybe they need different treatments. So we then can define by the dysregulated pathways, we can use maybe drugs to treat these children individually. And this is the future, I think, also for single cell-based approaches, which could be used as diagnostics also for rare diseases and not commonly occurring mutations. And the second point is, of course, heart failure is not heart failure. We have many different reasons for heart failure, and we have many different features. And by combining these biopsies, so taking small pieces of the heart with imaging, we can maybe learn what's wrong with the heart on a molecular level and combine this with imaging technologies. And later on, we only have to image because we know what the imaging tells us and what could be bad for the patient. And then again, we could find out what pathways are wrong and what could be potential therapeutic targets to treat heart failure patients, not all getting the same 6 or 8 drugs meanwhile, but having specific drugs for specific subsets. So it's a type of personalized medicine which we're aiming at. Mm-hmm. And finally, in the field of, I would say, a lot of chronic diseases like cardiovascular disease, but also maybe for BART, neurodegenerative disease, we have inflammation as a causal, or let's say contributing factor. And we could take the circulating blood cells, learning immensely how they are changed in these diseases. And this could be also a biomarker and diagnostic tool to see which of the patients may have an inflammation problem. So which patient may profit from an anti-inflammatory therapy, which patients have maybe a potential for getting more specific treatments with their different immune cells like T cell or monocytes or maybe other cells. And we already have drugs, but we don't know which drugs to use for which patient. And I think this will maybe a future for a consequence also of learning more of the cells of a body in the field of single cell technologies in general.
Mm-hmm. So what I'm getting from each one of you in your different areas of expertise is that this has a lot to do with multidisciplinarity and bringing a lot of different expertise, not just the clinicians, not just the molecular and cell biologists, but also the data scientists. You discussed about techniques that involve looking at single cells at the molecular level, also imaging, liquid biopsies, etc. So how is this happening now, or can happen in a better way in the future to help our understanding of disease and to help patients later? And maybe Nikolaus can give us his view on this also, because you mentioned Lifetime as an initiative. There are other such initiatives in other parts of the world, but how are you bringing together these teams and managing to, juggle all these different types of data and expertise?
Yeah, indeed, I think it's interdisciplinarity is key, and so nowadays in what we are trying to do, you need people who are experts in, let's say, machine learning to work in biology. And then you need the clinicians, then you need also the patients to be involved, and so on and so on. And so this is one of the reasons why we have also in Europe, we are behind Lifetime, so where we try to meet and marry these different disciplines. This, I think, is very important to develop certain locations where there's a specific push to break the boundaries between some of these disciplines. So we are proposing a number of, we call them cell centers, European cell centers in Europe, where people come together to really work between disciplines on cell-based medicine. I would like to say, maybe give a little bit of a concrete feeling for the data involved here. So the data that the single cell technologies are generating are quantitative data, which tell you about each cell, which genes are on and off, and actually in a quantitative way, and other things of other features of the genome can be probed as well. I will not go too much into this, but just to say that the Human Genome Project worked for 10 years or more on sequencing the human genome, which is 3 gigabases roughly. And each cell is reading this book of life in a different way. So a cancer cell does it very differently and is actually changing the book of life on its own. A neuron from Bart's Alzheimer world will read very differently the human genome compared to a heart cell from Stephanie Dimner's research. And so what is happening now is that we're getting a map between the molecules which are making the important decisions and functions in cells, but in tissues or in space. That means that we get information on the order of gigabytes per cell, but for many cells in parallel. So These are now could be 10,000 cells next year, probably 100,000, and soon enough 1 million cells. There are really no boundaries for this, in fact. And so, if you think about this, this is a absolutely enormous amount of data. And the beauty is it is not just enormous data, it is actually very, very meaningful data, because it tells you how the molecular makeup of cells is working to make cells communicate properly or improperly, or how cells are reacting to a drug or to other influences from the environment. So it's also a completely new way to really study the interaction between physiology in general and the molecular mechanisms that respond and adjust.
Mm-hmm. We actually have received an interesting question from the audience that relates to what you were just talking about, the different types of data, the sizes of the data sets as well. So what are your experience in each of your fields in terms of integrating data from different sources on a single cell, for example, mRNA data, proteins, DNA, spatial data as well? Maybe, Nikolaus, you could give us your perspective as the single cell expert first.
Okay. These are huge challenges for the data sciences. And I'm very, very happy that some of the brightest people in the machine learning fields and other data sciences fields have joined our quest to understand now really the molecular mechanisms in individual cells. I can only say it in this way. This is very complicated, and you need to, but it's doable, of course, because cells are also doing it. So you need to develop a lot of new methods. You need to think about mathematics, and computer science in very creative ways. So it is also important point maybe here is you cannot just outsource a problem to a data scientist and say, so here are the data, and now you run your methods. You really have to bring these people together, because a data scientist will constantly have to come up with innovative new methods how to integrate these kinds of data.
Mm-hmm. So a picture emerges where we can take samples from a patient, and then we can do different types of analyses on the cells of that patient that will inform mRNA levels or protein levels, or the actual types of cells that can be detected. Bart and also Stephanie spoke to some of the challenges of achieving this, because in the brain, obviously, we cannot get samples of the size that we need. In the heart, it is the same. So what are clinicians and biomedical researchers facing when you have to make these choices? You know, you have the ability to get some samples. How would you, what is the decision-making process in terms of how do we tackle this problem? What types of experiments do you do now, and analyses? Bart, can you tell us a little bit about this from the
It's really
neurobiology?
spectacular. And so you have, for instance, when a fantastic example is a cohort which is studied in Finland with people who get hydrocephalus. They need to have an operation to relieve a little bit of pressure in the brain. And 50% of these patients have also signatures of Alzheimer's disease. And so what these people, well, during the operation, it's not a problem to take a very small column of brain material. And so that has no effect on function, etc. And so this small biopsy is not sufficient to do any classical biochemistry. But for single cell biology, this is just more than enough. There are zillions of millions of cells in such a small piece. And so you can then very quickly dissociate the cells, or you can even use them as a structure and do spatial transcriptomics on these materials. And so it's completely changing the way we look at this stuff. So it's possible now because the scale, the number of the amount of material you need from patients is so small that you can really start to do that also for organs which you couldn't touch in the past, like the brain or like the heart. So I think we are really starting to see all these possibilities. So you could also do that when you install electrodes for the brain stimulation, or where you can take small samples. While you do that, you damage anyhow a little bit the brain while you put that electrode, and so the small sample you take is an order of magnitude less than what you do while you put the electrode there. So it's really changing the way we can look to the brain.
Stephanie, any thoughts about the heart and how you decide what samples to take for what experiments?
Decide is not the right question. I would say the question is what we can get. So we have actually two ways to get heart tissue, one of which is during operation, a little bit like Bart described, but in this case, you can get quite a significant amount of tissue when you implant left ventricular assist devices. These are types of artificial, or let's say, hearts, extracorporeal hearts, supporting the body if the heart is failing. And if this is done, there is a chunk of tissue coming out, which is otherwise thrown away, and we can get this. But this is end-stage material, meaning people which cannot live without this machine. And then, of course, this tissue is not what you would like to have. You would like to see the disease at an earlier stage. And this is quite difficult to get. So we can sometimes get biopsies, but we only get them if there is a medical indication, meaning if the physician thinks that there might be something wrong with the heart, but he doesn't know why the heart is not working. So for example, if you have a viral infection, so let's think about a COVID patient who has heart dysfunction. They want to know whether there is the virus attacking the heart or whether there's another problem. Then you might get a biopsy if the patient accepts and agrees that there will be one biopsy taken for science. Then this could be donated to our science. So this is what we're aiming at and hoping for. But still, it would be really nice to have a time course. Like in a basic scientist would like to see an early stage, mid stage, and late stage to see the kinetics. And I think one thing we can do and which we will do is to share data. So my vision is that we all put all of our data we have together with the clinical characteristics together. And if everybody does this, and then we may get a more global picture if we have enough patient material from all the different regions in the world, and then maybe we can get more complete pictures with the help of Nikolaus, who then puts all these data together and helps us to decipher maybe a type of time course, or at least say age and gender and treatment and subsets of heart failure. Then we may get more information.
So I'd like us to discuss the question of data and data sharing a little bit more in a minute. Before we move to that, Angelica, we've heard some of our panelists mention that in cancer, maybe getting material like samples from the tumors is not as big a challenge. Of course, it is somewhat of a challenge depending on the cancer type, but also on the disease stage, right? So when we have a lot of tumor material to test, the patient is in trouble. So how are the single cell technologies and cell-based approaches helping us to detect the disease earlier and hopefully diagnose patients earlier?
Well, also in the cancer field, it's a stepwise approach. So what we usually do when we diagnose cancer is to take a tiny biopsy, and then we have a lot of routine diagnostics to do in addition to routine pathology. And routine currently even involves at least panel sequencing for some parts of the patient's whole exome sequencing, whole genome sequencing. But then the biopsy material is gone, and we don't have enough material left for single cell technologies. This means that we have to first implement the technologies from samples that are derived from what we call second look surgeries, when we do the large operation resecting the tumor that has not responded to chemotherapy. But this also implies that these cells have already seen treatment, chemotherapy in general, and this treatment causes changes. And of course, we would like to learn from more informative, naive samples in the very beginning. So we have to first implement implement new technologies with material where we have sufficient material derived from these second look surgeries, and then try to narrow it down to single cell approaches so that we don't need so much material, and that it might be feasible to do it from the first biopsies. But also at the same time, we have a lot of efforts in analyzing blood samples, circulating tumor cells, but also cell-free DNA and RNA and exosomes that are circulating in the bloodstream of the patient, and trying to identify biomarkers in the blood that can be analyzed longitudinally in the clinical course of disease. And this field of liquid biopsies of blood sample testing by single cell technologies even now makes a lot of progress, and we already have biomarkers that we can follow in the blood and tell the cancer patient in between if the disease is responding well or not so well.
So we are, for some fields, we're on the cusp almost of expanding the applicability and the availability of some of these methods, right? You just mentioned liquid biopsy. There are even commercially available approaches that have been rolled out, which is amazing. And then we have in some other fields bigger bottlenecks, let's say, in terms of getting the samples and doing these analyses. But we also have the other side, where, as Stephanie and Nicholas also mentioned, with the ability to generate a huge amount of data, and then we have the challenge of integrating these data and also sharing the data, which is something that Stephanie touched on. So I'd like to hear your thoughts about this, also thinking of the bioethics side, because we're talking about patient data and we're talking about privacy matters. And also not just when it comes to the data sets, the single cell data sets, but maybe also to the patient specific models. You know, we have cell-based assays like organoids that can lead to ex vivo models of disease. What are your thoughts about this? Perhaps, Nikolaus, you could start in terms of the privacy and sharing aspects.
Yeah, first of all, the good news is that in Europe we now have a pretty good law that protects the patient rights. So that's called GDPR. And I think that is really an accomplishment because it makes sure that an individual patient owns the data that are ever generated from that patient, simply speaking. Now, this of course also has implications. You cannot just in the clinics do measurements and then send all of this somewhere else and do conclusions from it. This all has to go through a very, of course, complicated procedure that starts with the patient, but then goes to well, follow the guidelines to protect the GDPR. And then the problem comes that this is different from state to state in the European Union. And so you have to also work on setting up federated data systems. But the very good news is that we are not here to start this very complicated process now. There have been actually for a long time European efforts and working groups and organizations to set this up, that such a federated system will eventually come together. It's not really there yet, but I think it's the only way to really move forward, also as Stephanie indicated. And so I see this as an opportunity to just say these efforts are super important, and I hope that the European Commission and the national countries will support the people working on this to make this reality.
Mm-hmm. So thinking a little bit more about the broader availability of such methods and techniques for patients? Obviously, we're talking about methodology and technology that is very advanced. It's not available right now at every hospital or at every university. It's actually, things have changed a lot over the last 10 years, right? And things are changing rapidly, but we're not at a place yet where every patient has access to this. What are your thoughts on how fast we're progressing to at least generating some workflows where patients can benefit by having samples sent to centers, or doctors having more of an option in terms of how the samples of their patients are assessed? Maybe Angelica, you could talk to us about this from the cancer perspective.
I think the cancer field is kind of a blueprint for this, because in this field, single cell technologies and molecular characterization has started in a personalized medicine fashion. And at least at the university hospitals, at the large cancer centers, the comprehensive cancer centers all over the world, there are personalized medicine programs where at least advanced stage cancers that have no very good first line treatment option are included. And they are characterized molecularly, currently not by single cell technologies yet. This is just small patient cohorts where we apply this, but by bulk sequencing procedures, this is already done. And then the results are discussed between bioinformaticians and biologists. Prioritization algorithms are sorting the data and giving us as clinicians the input where drug targets are found and identified that make sense for targeting. And then all these results are discussed in so-called molecular tumor boards, where the clinical information of the patient's imaging information, the cause of disease, are matched with the molecular results. And then an interdisciplinary panel is deciding what is the best step for the patients, what is the best targeted molecular therapy or cell-based therapy that can be used for this individual profile and these molecular signatures that we find. So this is the status quo in comprehensive cancer centers, and we are trying to network and to implement outreach programs in the different countries, so that also patients that are treated not in these centers but in smaller hospitals or in private practices can get access to these options. And there are digital health solutions on the way, where a patient, for example, can find the website and provide his or her data to the website, and then gets feedback if it makes sense to send in biomaterial to a central address and stuff like that. So this is implemented in clinical routine, but it only starts, and I think it can serve as a blueprint also for other disease fields.
Mm-hmm.
But Alexia, can I say something also to this point?
Absolutely, yeah, of course. Please.
I think you asked a really important question, how these new devices, methods, and so on really will come to the patient. And I have to say that I view, or we in Lifetime view, the emergence and support of cell centers in several European locations as absolutely crucial for this. Because you need some critical mass to develop these kinds of methods and to make them really available. And you also need to really work together with innovation and companies to actually make them reality. And this is exceedingly difficult if everything is fragmented over all of Europe. So we think we need these places where certain technologies are with a particular critical mass and weight pushed forward and connected to innovation. So another point about this is that you cannot just create beautiful instruments that measure something. You also need to set standards. Otherwise, data that we want to share are not really comparable. protocols are not comparable, so you really need to set standards. It's super important so that we can really as a community move together. And this also must be done at particular places where really the critical mass is behind technology to be able to really define these standards in optimal ways and share them and provide also access to them.
So it sounds like from a European perspective, I'm European as well, so we have some very good ways to move forward in that direction because we're so interconnected in terms of our countries, but we also have these barriers of the different country rules and regulations that we have to overcome. So do you view this as a concerted effort to also educate, like one country in which some of these technologies might be more developed, educating colleagues in other countries, other clinicians, hospitals and different centers? I was addressing Nikolaus, but any one of you, of course, is welcome to answer this.
Yeah, I just think it's interesting. Sorry.
No, I totally agree with this. Of course, it's also meant to share and educate and provide access and disseminate and so on. But Bart, please go ahead.
I was just thinking about the UK perspective in this whole discussion, where I must say that in a certain way they are very strategically thinking. For instance, data sharing, etc., they try to organize that over their own country, trying to be a little bit at arm's length of the European Union. But they have really this vision of bringing all the clinical data together, making possible to mine them, to do early proof of concept of drug targets, etc. And so they have these centers for data science, Turing Institute, a couple of other efforts. They are trying to bring all their NHS data together and accessible for this exploitation. And so I think it's sometimes good to look a little bit how they are doing these things. I think that Europe should do the same, but then at the whole European level, and that's really the problem that we are still all separate countries with our own agenda. Mm-hmm. So the other point I wanted to make is that I'm very supportive of the vision of Nikolaus, that we need a couple of specialized cell centers in Europe which take the lead in this whole process. And I think that the European Union should do this and say, look, we put 5 or 6 or 10, the number is out in the air, where we get a real focus on developing these technologies for various diseases. So the center should be linked to hospitals. They should also be linked to the data centers. That can be a little bit more virtual, but I think we need this top-down approach to really get this forward.
Mm-hmm. So unfortunately, we only have a few more minutes, and this is a huge topic. So I will switch gears a little bit to another aspect of cell-based medicine that I think it's important to discuss. And this is also coming from one of the members of our audience. What about modeling disease with cell-based approaches? And I did mention organoids previously. This is something that has been well developed over the last few years in all your areas. Maybe Bart can start on this topic, given that you also mentioned the importance of the niche for understanding the disease.
Yes. So I think one of the biggest challenges we have in all these sporadic diseases is the genetic makeup of a human, the risk factors, the risk genes, so which you can bring together in a polygenic risk score. It's very difficult to study that in an animal model, and so we need good human models. And so the organoids is certainly a way forward. What we also do is a lot of chimeric mice, where we bring in human cells into a disease condition and then look what the effect is on the human cells. And I think this whole new philosophy around single cell analysis allows you to bring, I was just at a meeting about organoids, human organoids, and when you see how that allows, the single cell approaches allow you to extract all the information about all these different cell types, how they interact with each other, what pathways. This is really making so much more use of this type of models than ever before. And then at the same time, getting the information from our single cell analysis in humans, in patients, etc., which is always a little bit fragmentary, but we can overlap that information with the models which we are generating. And this is really changing, I say it the whole time, but it's really changing the way we look to, for instance, neurodegeneration, where we can then, I mentioned the cellular niche, which is for the cancer field, something evident, but for us, it's really a complete novel way to look to these disorders.
Stephanie, can such models help us in understanding and modeling cardiovascular disease?
I think that I fully agree with Bart. So I think this is exactly what we also envision for cardiovascular disease, so organoid models. And I think the combination of primary cells, for example, circulating blood cells, and then putting it in organoids and having this together, this is really very helpful. And it's also important because I also see that particularly these age-associated diseases, it's very difficult to mirror them in mice models. And we need human model systems to work on this.
Angelika, Bart also mentioned that the tumor niche is very well established in the cancer field, of course. Of course, we also know of the challenges there, and getting an organoid model that is faithful to the tumor setting, the microenvironment, is very challenging. Any thoughts on this and how this field is developing to help patients?
Yeah, certainly we need to put efforts into these models and make them less artificial. And it's not enough to just cultivate 3D cells derived from a patient's tumor, but we have to model the niche as well, meaning that we have to bring in vasculature vessels into these organoid models. We have to bring in immune cells, stromal cells that are around a tumor. And of course, a lot of developments like this are underway. We even deal with 3D human on chip models that mimic different organ systems. But still, all of the models around are not the perfect, what I like to call avatar for a patient. And we need to put in more efforts in developing better models that help us to predict responses to certain treatments, because we're living in a fascinating time, and a lot of targeted drugs, a lot of immunotherapies are developed, and each individual patient, each individual tumor is different. So we need to know, hopefully upfront, which kind of therapy is the best choice for every patient. And if we want to find out about this, we need an avatar model of each patient, a patient-derived model that we can use for numerous drug testing approaches. And of course, in this way, it has to be faithful, because otherwise we are predicting the wrong thing. And the challenge definitely is to bring in all these different cell populations in a meaningful way, that they can really interact in a model as they do in a living organism.
Maybe I can add something. I think what is important is that we can use the single cell sequencing data, which we have obtained and will obtain with biopsies or tissue material, and then see whether our organoid model really mirrors what we have in the patient. So we can use these data from patients to building our organoids and other tissue culture models much more realistic. I think this is one of the beauties I think also these data have, and which could be a blueprint in the end for our tissue models. I think this is one advantage which we can gain from the data which we have from the patients as well.
Absolutely. We have an audience question for Nicolas specifically. Following diagnostic modeling efforts, how does the Lifetime Initiative envision disease intervention and remodeling of dysfunctional cells? Which I think actually could be a question for all of you. But
okay, so once you can really, based on molecular mechanisms, predict where a cell in a disease is, so to speak, moving the disease trajectory or the trajectory of the tissue, you then also can have it in your hand to systematically interfere in the system, because you know the molecular mechanisms driving the system. So I think the approach will give us a huge number of new drug targets to interfere with the system and to bring the cells back to health. I would like to say something just because also time is really coming to an end regarding the organoids. It should be clear to everybody there was just a Nobel Prize for Jane Doudna and Emmanuel Charpentier about the ability, they invented a way to very precisely interfere or edit DNA or hereditary molecules. By the way, you can also, with this technology, target gene expression, which is also extremely helpful because you can do it without changing the genome. And you have, with this technology alone, a unbelievably powerful method to unleash on the organoids for a specific person's disease. So you can really not only look at it, but you can also perturb the system to test your hypothesis, to test new drugs, which ones are really the best, which work best, which ones are the least toxic and the most specific, and so on.
Thank you very much. Unfortunately, I'm very conscious of the clock and our time is coming to an end. So I would just like to thank all of you for participating in this discussion. What I am coming away with from this discussion is definitely that there is huge potential from cell-based methodologies to cell-based therapies. And a lot of this will come from collaboration and sharing knowledge and sharing data, always having the patients in mind and how best to help them and serve their interests. So again, a huge thank you for participating in this table discussion today. And thanks also to our audience. Please do keep an eye out for the following roundtable discussions that will happen tomorrow. And also, as I mentioned previously, you can catch our daily highlights every day at 1 p.m. Central European Time. Thank you all very much.
Thank you, Alexia. Thanks, everybody.
Thank you.
Bye-bye.